Hello everyone, Mr. Linder here. Let's talk about protein production. So proteins run the day-to-day activities of cells, and therefore protein synthesis is critical to our cellular function. The power of proteins comes from the tremendous variability and specificity of those proteins. There are 20 genetically coded for amino acids that can be rearranged into thousands of different proteins with different functions. Changing just one amino acid during protein synthesis could alter the function of a protein and lead to pathology. It wasn't until the 1960s that biologists were able to figure out how the DNA code could actually code for 20 different amino acids. It turns out that bases from our DNA in triplet code would be the code for amino acids. So we can take the A, T, C, and G bases, and we can look at bases in a sequence of 3 and figure out what amino acids are coded for by that sequence. There are 64 possible codons. The 64 possible codons comes from the fact that there are 4 bases in triplet code. So 4 to the 3rd power is 64. But since there's only 20 genetically coded for amino acids, some of those codons are repetitive for the same amino acid, or they're used for some other code. So what are the triplet codes? Well, the triplet codes are called codons, and they're on the screen. And you can see some are UUU, or CCC, or CAA, or AGA. So there's obviously variety to the code. There are 64 codons, and we use those to code for the various amino acids, or again, other things that we need for the processes of transcription. In the 64 codons, one of the codes, AUG, is actually used as the start codon, but it also codes for the amino acid methionine. So the 3 letters are the abbreviation for the particular amino acid. So AUG is a start codon, but it also codes for the amino acid methionine. There are 3 codons that are stop codons: UAA, UAG, and UGA. They all code for stop in the process of transcription. The other 60 codons code for the various amino acids. And these other 19 amino acids, you'll see that there are some codes that repeat for the particular amino acid. So for example, leucine has 6 codons. 1, 2, 3, 4, 5, 6. And there are others like proline that have multiple codons for it, threonine, alanine, and so forth. So we use sometimes repetition in the code to protect against mutations that potentially could happen. So sometimes when you change just one base, that doesn't necessarily mean that the amino acid will be different. If you have CCC and CCA, you're still going to get the same amino acid, proline. And so that can protect against some mutations and therefore protect some of our proteins so we don't have pathologies that happen due to changes in the genetic code and changes in the protein structure. So how do you unlock the DNA code? How does the process work where you go from the bases, so where you go from having A's and T's and C's and G's, how do you get from a code like A, C, T, A, C, C, G, G, and so forth? How do you get from that code to actually producing a protein? What are the processes that we need to go through? Well, we need to take a look at unlocking the DNA code. So what we really need to do is we need to be able to produce RNA from the DNA, and we need to be able to produce a protein from the RNA that we produce. And this really is the secret of life if you think about it. DNA to RNA is what we call transcription. And then RNA to protein is what we refer to as translation. Now, there are other processes that we're going to be taking a look at, but these are the two main ones that when you think of prokaryotic life, bacteria, when you think of plants, animals, fungi, DNA to RNA to protein is the cellular secret of life. So what has to happen? What are some of the steps when you go from a gene to the production of a protein? What are some things that we need to take a look at? So number one, there's a process of gene activation. So a gene must be activated so that its code can be read. Some genes are what we call constitutively active, meaning they are continuously being read and converted into RNA. So they're essentially always on. And then you have what are called regulated genes, and these regulated genes have to be induced or repressed. So if they're repressed, they're off. If a gene is induced, then we're saying that we're turning that gene on so that we can go through the process of transcription. So the first thing to look at is this idea of gene activation. The second thing to take a look at is once a gene is active, what types of RNA can you produce? And it turns out we produce predominantly 3 types of RNA, at least that's what we're going to focus on. There are other versions of RNA, but the 3 main ones to think about are messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA, we're able to produce that particular type of RNA, so mRNA, because we use a particular enzyme called RNA polymerase, so RNA poly version 2. So when we use RNA polymerase version 2, that particular enzyme is used to read the genetic code and produce messenger RNA. Transfer RNA, we use RNA polymerase version 3, and that particular enzyme is used to make transfer RNA. And then when we need to make ribosomal RNA, we use RNA polymerase version 1. So depending on what enzyme we use in the transcription process, we can get different types of RNA. Messenger RNA, transfer RNA, and ribosomal RNA. And all 3 of those are important in protein production. The third thing to take a look at is messenger RNA processing. mRNA processing takes place in the nucleus, so we're still in the nucleus. All of this in blue is in the nucleus. And mRNA processing really comes in 2 forms. There's interference, which essentially is going to turn off the messenger RNA. And then there's alternative splicing, which is going to give us the active form of messenger RNA. So mRNA processing is all about giving us the active form of messenger RNA. Once you have active messenger RNA after it's been spliced, it can leave the nucleus and it can go out into the cytosol and it can interact with ribosomes. So messenger RNA will be processed we can process or read messenger RNA once it leaves the nucleus. And we do this through a mechanism called translation. So translation is where we're going to interact the messenger RNA with a ribosome, and then we're going to use transfer RNA to look at the code along the messenger RNA and figure out which amino acids go in what position. Once you have the amino acids in the appropriate position, you have a polypeptide structure, which is essentially on its way to becoming a fully functional protein once it folds and it creates the appropriate shape. But oftentimes we have a 5th process, which is called post-translational modification. Once translation takes place, new proteins have to fold. There might be enzymatic cleavage that takes place. We might add additional functional groups like sugars or lipids or phosphates, or we may even combine multiple proteins together to make a new protein that will then go on and do work within the cell. So we've got a lot to unpack here and take a look at as we're trying to figure out how to produce a protein so that proteins then can go on and do work. So really the first thing to look at when you really start to unpack this is this idea of transcription. How do you get from DNA to RNA? So the process of getting from DNA to RNA is this process of transcription. And we're going to focus mainly on the RNA polymerase version 2 enzyme because that's the one that allows us to produce messenger RNA. So when you see RNA polymerase in these diagrams, you want to understand that we're really talking about RNA polymerase 2 because we're trying to make messenger RNA. So the first steps in producing a protein actually take place within the nucleus. And transcription is the process by which we read the DNA and we use the template bases to produce messenger RNA or an RNA structure. And to do that, we need enzymes like RNA polymerase number 2. To initiate transcription, we need to activate what's called a promoter region. So in order to get from the DNA, this is the DNA right here, in order to get from the DNA all the way to where we have a messenger RNA strand, we have to first activate the DNA at a particular location. So we want to read a section of DNA, we want to read the gene, but we're going to have to activate that section by interacting with a promoter region. And that promoter region is where we're going to bind regulatory proteins and bring in our RNA polymerase so that we can actually start the transcriptional process. So the promoter is in front of the transcribed gene. And then regulatory proteins called transcriptional factors are going to interact with the DNA to activate the promoter so that RNA polymerase can come in and bind, and then we can actually read the gene. So this is the RNA polymerase in this diagram. You can see that the DNA is already opening, so you can imagine that transcriptional factors have already come in here and activated the promoter, so the enzyme can come in and bind, and we can start reading the code, right? We need to read the gene in order to produce the messenger RNA. So what does this look like? So if you want to look at it in a more enhanced view, we have something like this. So what is the process of transcription? Well, number one, transcriptional factors, and there's lots of different kinds of coactivators and repressors and all kinds of different things. But transcriptional factors need to come in and interact with the promoter. So this region of DNA, so this strand right here, this is the DNA. This region of DNA is the promoter region. So this is the promoter region. And then this is the gene going this way that we're going to transcribe. We're going to read that gene. So transcriptional factors, right, all kinds of different transcriptional factors, have to come in and interact with the promoter region so that we can bring in the RNA polymerase enzyme. So this is the enzyme right here. How do the transcriptional factors find the promoter region? Well, promoter regions typically have repetitive sequences. So TA, 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 repetitive sequences for these transcriptional factors to find. Sometimes you'll see CAAT as a repetitive sequence, or you'll see GC repetitions. And those repetitions are the code within the DNA for transcriptional factors to come in and interact with that portion of the DNA. Once the addition of the transcriptional factors happens, this allows for the RNA polymerase enzyme to come in and bind. So you could think of that as step number 2, and that forms something called the initiation complex. So we have the sort of non-open DNA. We need to get RNA polymerase to come in and bind to the DNA, but the only way that's going to happen is if the transcriptional factors come in and interact with the promoter region. So we got to bind to the promoter before we can read the actual target gene. You know, what is our target gene? What protein are we trying to get from this? Because a gene essentially codes for protein. So we got to get the transcriptional factors in there before the RNA polymerase can come in. Once you have the RNA polymerase in place, you have your initiation complex. So this is a fairly complex diagram, but I wanted to show you that here's the DNA up at the top, okay? And we have the promoter region with its repetitive sequences, CAT box, GC box, TATA box. Okay? Transcriptional factors, these little jelly bean looking things, transcriptional factors coming in and binding to the promoter so that we can bring in the RNA polymerase version 2. So you can see it listed right there. Okay, RNA polymerase version 2. And now it's attached to the DNA. So right here, that RNA polymerase is attached to the promoter region. You've got your transcriptional factors that help to bring it in. And now we can open up the DNA and actually read the target gene. So the target gene is down here. All right, we're going to read this section. And as the RNA polymerase moves down the DNA, it's putting in the appropriate RNA bases. So RNA polymerase is going to read the gene, and it's going to put in the RNA bases. So what are the RNA bases? Well, in RNA, we use A, we use C, we use G, but we don't use T. We use U. Okay, we use uracil. So adenine, cytosine, guanine, and uracil are the RNA bases. So RNA polymerase is unwinding that DNA. It's reading the code, and it's going to put in the appropriate bases. So the DNA is the template for the RNA code. And so we have to put in the complementary RNA bases. So for example, if you had a DNA code, A, G, T, A, C, something like that, if this was the DNA code, then the RNA code would be U, because A and U go together, uracil goes with adenine, C, A, U, and G. So you'd put in the complementary code. And every 3 bases is called a codon. And if you want to know what that codon codes for, you go back and look at that 64 codon coding sheet, and you can look up UCA and figure out what that is actually coding for. Once the complementary bases are in place, the RNA polymerase will keep reading until it reaches a stop codon. Once you have a stop codon, then you'll have what's called the termination complex. So the portion in the middle is called the elongation complex. That's when you're moving along the DNA and putting in the right RNA bases. Right? So we're adding in the RNA bases. You're elongating the messenger RNA. When you get to the stop code, you've reached what we call the termination complex. And so things are going to separate from each other. RNA polymerase is going to come off, the messenger RNA is going to come off, and then we can go into messenger RNA splicing. So if the newly formed RNA is messenger RNA, it'll go through messenger RNA splicing. So what is messenger RNA splicing? So after this is just another diagram for creating the initiation complex. So if you want to take a look at this one, you can see the same stuff. DNA, we have transcriptional factors interacting with the promoter. We bring in the appropriate enzyme, and this is our initiation complex. So this initiation complex is on the promoter region, and it's there so that the enzyme gets in the right spot, and then we can read the target gene. If you have messenger RNA that's produced from transcription, then we need to go through processing. So let's get to a new diagram here. So if you've taken the DNA, so here we have the DNA, and you've produced RNA, specifically messenger RNA, and notice U's go with A, G's goes with C, UA, CG, and so forth. If you've produced messenger RNA, then we need to go through the processing of messenger RNA. That processing comes in two forms. It's either interference, which is going to turn the messenger RNA off, or it's an mRNA splicing that's going to give us the functional messenger RNA. So RNA interference, it looks something like this. We're going to just simplify it and say that RNA interference is a way to turn off the RNA. It probably evolved from a mechanism where cells were needing to block RNA viruses, and so we developed a way to chop up the RNA. And so now we can actually use it in our cells to basically break down messenger RNA when it's no longer needed. The main enzyme to do that is the dicer enzyme. So here we have the dicer enzyme. So the dicer enzyme can chop up the messenger RNA and degrade it so that it's off, it's non-functional. So think of RNA interference as a way to turn off the messenger RNA. Alternative splicing is where we want to process the messenger RNA into a functional piece of messenger RNA. And this involves removing sections of RNA and then splicing new sections together. In alternative splicing, we use enzymes known as SNRPs. So SNRNPSs, SNRPs, small nuclear ribonucleoproteins, are the enzymes that we use for processing messenger RNA. So in this diagram, we have DNA that has become messenger RNA, unprocessed messenger RNA, and we're going to use the enzyme SNRPs to chop out certain pieces and splice together the remaining pieces. The pieces that we cut out are known as introns. So introns are the pieces that we remove. Exons are the pieces that we splice together. So exons get spliced together, and they become the new messenger RNA. Now notice in this general example, in the first example here, section A of the unprocessed messenger RNA was an intron. Okay, in this first part, the SNRPs came in and cut out section A, and they also cut out C and so forth, and they spliced together B, D, E, F, and H. So section B is an exon, section A is an intron. But notice in a different example here, section B became an intron, section A was an exon. And the reason I point that out is so that you understand that the sections of unprocessed messenger RNA are not fixed sections. They're not always going to become one thing. So section A is not always going to be an intron. It was an intron in this first example, but it was an exon in the second example. All this is a way for us to chop up the code and rearrange it so we can produce different types of proteins. What does that ultimately mean for us genetically? It means that this gene can actually code for multiple proteins. You can get multiple proteins from one gene. So if the genome is 25,000 genes, that doesn't mean that we can only get 25,000 proteins. Turns out we have over 100,000 proteins. So one gene can actually code for multiple proteins, and this is a great example of how that happens. These SNRP enzymes are chopping up the messenger RNA and rearranging it so that we can produce new types of proteins. So introns are the parts that got cut out, exons are the parts that put together. And this gives us an advantage so that we can produce multiple types of proteins even from one single gene. Here's another just simplified example. So here we have the unprocessed or pre-messenger RNA. Here's a section that's going to be chopped out. So if the section is chopped out by the SNRP, then it becomes the intron. And the sections left behind are the exons. So this exon, this is the first exon, this is the second exon. They're being brought together. So here we can see exon 1 is now continuous with exon 2, and you've taken out this section of code. And so now you have a new piece of messenger RNA that can then go on and be translated. So we're not translating the pre-messenger RNA, we're translating the processed messenger RNA after it's gone through mRNA processing. Here's another diagram just to show you the complexity. So these are various SNRP enzymes, U1, U2, U5, U6, U4. So these proteins form this complex called a spliceosome, and the spliceosome is chopping out the messenger RNA that will become the intron, and then the exons, exon 1, exon 2, can be spliced together. So here's exon 1 and exon 2. And so now we have this new messenger RNA that can be translated. So we go from transcription, then we go through mRNA processing, and then once we have processed the messenger RNA and created this functional messenger RNA, it's going to go out into the cytosol and be translated at the ribosome. So translation is the next process to take a look at. So here we have this summary diagram where you have DNA in the nucleus. You've produced a messenger RNA structure. It's gone through messenger RNA processing with the SNRPs. And now it's leaving and going out into the cytosol to interact with a ribosome. So these are the ribosomal pieces. And the messenger RNA is interacting with the ribosome so we can start the process of translation. So what is the process of translation? Translation is actually reading the codons. So taking those 3 base sections and bringing in the appropriate amino acids. So UUC is coding for phenylalanine. UGG is coding for tryptophan. AAA, this codon, is coding for lysine. And so we're bringing in the appropriate amino acids so that the polypeptide can grow and you can create the protein that is going to be used by our cells, by our tissues, by our organs. So the process of translation is basically an interaction of messenger RNA, ribosomal RNA, and transfer RNA. So the processed mRNA leaves the nucleus, goes out to the ribosomes where translation can take place. What's interesting is translation can actually take place on a polyribosomal structure, which is kind of cool. So this is the first ribosome reading the messenger RNA. So that'll create the first protein. But if a second ribosome, attaches, okay, then that's going to start making the same protein. So this is the second protein, but they're identical to each other because each translation is reading the same messenger RNA. So these ribosomes are attaching to the same messenger RNA. So this third ribosome is going to allow us to make the third protein. This fourth ribosome allows us to make the fourth copy of the protein. So polyribosomal structures are interesting because you can make a lot of the same protein from one single messenger RNA. So how does translation proceed? So Basically, messenger RNA has to first bind with the ribosome. Once it binds with the ribosome, then we have to be able to read the code. Now, how does the messenger RNA bind with the ribosome? Well, the messenger RNA interacts with ribosomal RNA. So the reason the ribosome and the messenger RNA can bind with each other is because a ribosome has ribosomal RNA in it. Once the messenger RNA and ribosomal RNA are linked, and you have the ribosome attached to the messenger RNA, then transfer RNA can start coming in and bring the appropriate amino acids. So I like this diagram because it shows you all the various codes. So here we have the DNA code that we started with, and through transcription, we were able to get the RNA code. So this is now the messenger RNA code, and this first codon, AUG, you might recognize it, that's the start codon. But you also know that the start codon is the code for the amino acid methionine. So this structure right here is the transfer RNA, and the transfer RNA has to be able to read the codon in order to bring the appropriate amino acid. So transfer RNAs have their own 3-base sequence called an anticodon. So the anticodon is going to link up with the codon, and we're going to bring the appropriate amino acid. So in this case, UAC is the anticodon for AUG. But we read the AUG off the table to know that we're bringing methionine. The next codon is CCA, and if you read the code table, you know that CCA codes for proline. So the anticodon for transfer RNA is going to be this GGU. So on the transfer RNA, there's a GGU sequence. It's going to link up with CCA, and we're going to bring in proline. And so now methionine and proline can form a bond with each other, so they can peptide bond together. And now we have a dipeptide. And then once we read the third codon, we can get another peptide bond, and now we have a tripeptide. And you can see what happens from there, quaternary and so forth. And so what you end up is you end up growing the polypeptide, where anticodons link up with codons, and we bring in the appropriate amino acids. So translation is just going to continue as long as there's a codon to be read, and we can bring in the appropriate amino acids. The opposite side from the anticodon is the coding sequence to link up with the amino acid. So there's a code on this end of the transfer RNA where the amino acid is going to link to. So transfer RNA is interesting because it has that anticodon sequence, but it also has the amino acid sequence. And so the appropriate amino acid gets put in that position. So polypeptides continue to grow until you reach a stop codon. Once you get to a stop codon, then everything can separate. Messenger RNA can separate from the ribosomes, the polypeptide is separating, and then it can start going through post-translational modification. When you have an empty transfer RNA, so here's what transfer RNA looks like. So here's the anticodon, and then here's the amino acid accepting region. When you don't have an amino acid attached, you have to put the amino acid on here. Okay, and so we have an enzyme that actually can reactivate the transfer RNA once it's been used. So in this scenario right here, once a transfer RNA drops off its amino acid, it needs to go through essentially a recharge process. So the anticodon has read the codon, it's left the appropriate amino acid, and now it leaves the ribosome, and then it's got to be recharged by an enzyme. That enzyme is an aminoacyl tRNA synthetase enzyme. It's a ligase type of enzyme that uses the transfer RNA, the amino acid, and ATP in the active site in order to recharge the transfer RNA. And so it looks like this. So here we have transfer RNA that needs to be recharged. This is the enzyme. So this is the aminoacyl tRNA synthetase. We got to bring in the ATP, we got to bring in the amino acid into the active site, and then we have to bring in the empty transfer RNA. And with the empty transfer RNA, we can combine the amino acid where it needs to go on the amino acid sequence. The anticodon is down here. That's the anticodon. We know that's going to link up with a codon eventually. But now what we have is a recharged transfer RNA. We've got the amino acid reattached to the tRNA. So now it can go be used in translation again. So translation just keeps going and going and going until you reach that stop codon. Once you have a completed protein, ribosomes can separate, messenger RNA can separate. It could be read again. We could use it again until it goes through mRNA interference, until it gets processed and broken down or degraded. Then it can be read again. But that completed protein can go through post-translational modification. So post-trans, after translation, post-translational modification, we can then fold it and add things to it, and then it can go off and do its work as a protein within the cell. How do proteins know where to go? So last couple things here, protein sorting. Once you make a protein, how does it end up in the right place? So protein sorting involves proteins getting where they need to go within the cell. And sorting of proteins within a cell involves having what's called a leader sequence or not having a leader sequence. So in this particular diagram, this particular this particular protein has a leader sequence. If it didn't have a leader sequence, it would be destined for a different location. So the leader sequence is typically about the first 30 amino acids of the polypeptide. And what it does is it signals where the protein is supposed to go. So the leader sequence directs the protein to a particular organelle. So some leader sequences might allow the protein to interact with a mitochondria. Some might allow it to interact with an endoplasmic reticulum, etc., etc. If it doesn't have a leader sequence, then it will remain in the cytosol. So if you produce a protein without a leader sequence, it'll become a protein in the cytosol of the cell. So we'll assume these proteins did not have leader sequences, and so they folded and became proteins within the cytosol. But in this scenario, because there is a leader sequence, this destined that protein to end up inside the endoplasmic reticulum. This leader sequence was for the ER. Once it's in the ER, we can go through the folding process. Notice a sugar is being added, so that's part of post-translational modification. This now makes this a glycoprotein, and we can see that continued three-dimensional folding to give us the functional protein. So cytosolic proteins are not going to have a leader sequence. Those proteins that do have a leader sequence are destined for particular organelles within the cell. What are some common types of post-translational modification? Oops, I'll just leave this picture up here. Some common types of post-translational modification would be to fold into the tertiary structure. So seeing all the folding that's taking place, that 3D structure. Cross-linking, so where you get like disulfide bonds, where you take proteins and they begin to covalently bond together, and you get other structures that are formed. Chopping up proteins, so maybe enzymes or hormones that get chopped up so that they become functional, like trypsinogen becomes trypsin or pepsinogen becomes pepsin. modifying palm C, which is a large protein that is used to create various types of hormones. So translating it into multiple different types, cleaving it and creating various fragments. So lots of different examples. Adding sugars, adding lipids, that's a post-translational modification. So making that glycoprotein or adding a lipid, making a lipoprotein. And then also creating polymetric proteins where you get things like hemoglobin, where you have, a protein and then another protein, and they're linked together, and then another protein, because hemoglobin's a quaternary protein. So you link all four of these globin structures together, and they form a much larger structure. So the quaternary structure of hemoglobin is a great example of these polymetric kinds of structures. The last thing to consider with proteins is, what do you do with proteins when they're no longer useful and you need to get rid of them? So misfolded proteins, proteins that have outlived their usefulness, need to be degraded, and they need to go to a location called a proteasome. And the proteasome then has enzymes to degrade the protein. So in this diagram, this structure is the proteasome. And so proteasome. And the substrate that you're trying to get rid of, so this guy over here, this substrate is the protein. So we've got a protein that has outlived its usefulness, and we need to get rid of that protein. In order to do that, we need to send it over to the proteasome to be degraded. So somehow we have to tag that protein so we know that that's appropriate for its degradation. So that protein has to go through this process of ubiquination. So when you see UB on this diagram, that is the ubiquitin molecule. So ubiquitin is added to the substrate so that we're tagging it so we know that it should be degraded. It'd be similar to going through your closet and tagging clothes that you're trying to get rid of. So you put a particular tag on it so you know those are the ones to get rid of and the other clothes are left behind. So we're tagging the protein. So here's the substrate protein. We're actually tagging it multiple times. So you can see ubiquitin, ubiquitin, ubiquitin. We're tagging it multiple times, and then we're sending the substrate and the ubiquitin tag over to the proteasome so that the substrate can become degraded. So these are the degraded particles of that protein. So the protein gets tagged, and then it goes to the proteasome, where it goes through a degradation process. I'll show you another diagram just so you can see this. So in this one, here we have the protein that needs to be degraded. So it's outlived its usefulness. Here we see the ubiquitin tag. This is the tag that's being added to the protein. Notice that it happens multiple times, so multiple cycles of ubiquination. And then we send it to the proteasome. So this is the proteasome structure. Here it is again down here. This is the proteasome structure. And the proteasome is where you have enzymes that are used to degrade the proteins that need to be broken down. This whole process is important because it prevents the cell from breaking down proteins that are still needed by the cell. So we tag proteins that need to be degraded, and we send them to a proteasome, and it protects the cell from breaking down things that shouldn't be broken down. So there's a lot of information on proteins, everything from transcription to messenger RNA splicing to translation to how proteins get to where they're supposed to go, and even down to protein degradation. I hope that helps. Take care.